Welcome everyone to our third MBIOS Research Insider sections in 2022. I hope everyone is staying safe and having good time in this weekend. We are excited to bring in more brilliant Malaysian scientists this year to continue their insight. To those who are new to Research Insider, welcome. In brief, Research Insider provides a chance to everyone to discover talented Malaysian bioscience researcher and to connect with them. Scientists will discuss in Research Insider, scientists will discuss about their works, their experience in science, and how they got to where they are now. These conversations will be accessible to everyone and are directed at non-specialists. Research Insider is also live streamed on MBIOS Facebook page. So there will be a Q&A section at the end where we will take questions from the audience and everyone can engage in the conversations together. So if you are interested in science or wish to learn about topic outside of your area of expertise, don't forget to tune in to Research Insiders every month and prepare to chat with scientists and learning something new. So let me introduce myself. I'm your host today. I'm Ong Kwan Hong, a PhD student in University of Malaya. My expertise in marine biotechnology. And a reminder to all the audience, feel free to post questions in the chat box at any point during Research Insiders. You can post it in the Zoom meetings or you can post it in the Facebook chat box as well. Also, if you look at the chat box now, there will be a feedback form in the chat. We are welcome and value your feedbacks. So our scientist of today is Dr. Jun Hoe, who is a bioinformatics project manager and at Quantitative Biology Centre, University of Tuebingen. His daily work includes provide project management, bioinformatics data analysis for various aspects of comparative genomics, such as RNA sequencing, single-cell RNA sequencing, different expressions, and phylogenomics. Without further ado, let's us welcome Dr. Jun Hoe. Doctor, the stage is yours. Hi, Dr. Jun Hoe. Dr. Jun Hoe, I think your mic is muted. Oh. We can't hear you. No, we can't hear you.
Can you guys hear me now?
Yeah, we can hear you now.
Yeah, sorry, I'm not sure what's happening. It was fine just a few minutes ago.
Yeah, that's what I'm curious as well.
Yeah, let's try again.
Yeah, sure.
Il y a beaucoup de placards dans la cuisine.
Right, thanks a lot for the introduction. So I'll give a talk. So today I'll share a little bit about what I've done previously and also a bit of my experience. So I just call it as biology in the era of big data, role of bioinformatics and data science. So yeah, feel free to ask some questions later. And I think this talk might be a little bit different from previous talks or might be what you expect from a typical biologist. So here's a little bit of my background. So I did my bachelor's of science.
Hi, Dr. Jun Hoe. Hi, Jun-Hoe. I think the voice lost again.
Can you hear me now? I'm not sure where some
Yeah,
people.
yeah. Probably it's connections. Are you using like
Yeah, I'm also
the?
with my mic.
Yeah,
I just changed. I used my default laptop mic now. Hopefully it's better.
yeah, it's
Right.
better now.
Yep. So anyway, I did my background. I did my degree in zoology with honors at UCAM. So the title of the degree is actually a bit misleading because it's actually more on molecular biology or biotechnology on animals. So actually a lot of lab work like DNA extraction, a lot of DNA stuff. And for my final year project, it was titled Molecular Phylogenetics in Sea Base. I'll explain a little bit later what phylogenetics is. But basically, here is where I start getting introduced to bioinformatics, or a combination of biology and informatics. So a more data analysis or method research on the computer instead of the wet lab, where you just do like DNA extraction, cloning, and so on. After that, I continued on with my master's in molecular biology at UCAM as well. And here I shifted a little bit more towards mathematics, so half on wet lab or lab work and 50% more on the computational analysis. And during the time as well, between 2009 and 2012, I did 2 short internships at the Center for Genomic Regulation in Barcelona and also the University of Aberdeen. I spent about 3 months at each institute, and the projects are related to molecular phylogenetics of ferritin, and both these institutes, I do a lot more bioinformatics analysis, just mostly on computational work. And let me now give you a short premiere what exactly is molecular phylogenetics is. So on the left, basically what you have is previously the species tree or the evolutionary relationships between different species. And in the past, people just do that using physical traits, like you examine that bony skeletons, pollens, and sort of divide between the species. That's what people usually do in the past. But these days, with tons of genome information and so on, we build what's called a phylogenetic tree, which is mostly based on DNA information, or based on gene and genome information. And this is more using computation analysis. And so this is done to establish the evolutionary relationships between different species, and particular applications like these days, for example, like COVID or SARS, for example, virus can even track how virus spread and evolve. So for example, for example, I think it was COVID is spread from bat or silver cat. So on this, how you track them, where you examine the relationships between the evolution of the gene itself. So anyway, this is a continuation of the background. So in 2012, I did my PhD in biology at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden. Also linked to the Max Planck of Physical of Complex Systems. They're both institutes are linked together. And technically, because these research institutes, I have to be enrolled at the Technical University of Dresden to get a degree. And actually, I've only been there twice to register and to collect my degree. But anyway, at this point, I fully moved towards computational, so I don't do any more wet lab. So I have to brush up my computing skills. I have to work with Python, Bash, and R. So I'm not sure if you're familiar with computer languages like C, C++, and so on. So I don't know C, so I usually work with Python and Bash and terminal. After that, I did a postdoc in Uppsala University in Sweden, where instead of work on mammalian genomes, my work is now on metagenomes of microbes. And then after that, did a short postdoc in a different group back at the MPI CBG, where I analyzed the genomes of flatworms. And then after that, COVID happened, and I was being stuck for a while. So then I managed to find a new position at the Quantitative Biology Centre at the University of Tuebingen, which is where I am currently. So for today's talk, I'll talk a little bit about my PhD project, and then also of what I'm currently doing at Tuebingen. So here's I'll start with my PhD project, so which is titled "The Detecting Signatures of Convergent Evolution Genome-wide," and this is the front of the front side of the institute. You see these green grills. Here's some pictures around the office institute. Actually, because I work on computational, so this is basically my office, and this is some of my colleagues hanging out. I think it's after a party or something. And this on the right is also the office. And I think this was where we were labeling our things because we're supposed to move to a different office. Yeah, and this is the front of the institute, which is actually quite nice to have lunch at in spring or summer. So I'll need to give a little bit of introduction to some biological concepts before I can understand my project a little better. So hopefully we find for you guys. So yeah. So I'll need to give a little bit of Is someone speaking or something, a question or something? Okay. Anyway, so I need to explain what are genotypes and phenotypes. So genotypes is the genetic makeup of an organism, or like DNA. And for this purpose of this talk, I'll just simply refer to it as genes. And phenotype refers to the physical or biochemical characteristics of an individual organism. For example, things like height, eye color, blood type, and so on. So the simple message is that genotypes codes for phenotype. And phenotypes are mostly determined by genes, mostly here because I'm not talking about environmental factors like lifestyle and diet for the purpose of this talk. But simple phenotypes like the ability to taste PTC, which is a bitter compound, is controlled by just one gene. But other phenotypes, for example, hearing, like deafness in humans, for example, is linked to over 40 genes. So among the reasons why we sequence genomes and we study genes is we want to know, for example, what are the genes involved in a particular phenotype. And the old way or the previous way to do it is to test every gene one by one, which is a very difficult task because humans, for example, we have over 30,000 genes. And even then, it's also complicated because, as you see, simple phenotype like hearing, for example, can be linked to over 40 genes. So a lot of these genes work in a network. So even if you identify one gene, you still don't exactly know how the genes affect the phenotype. So I'll try to walk through this very quickly as well, as in a simple manner. So genes contain information to create proteins. So if you look at this fancy picture on the left, this is how you always assume a DNA and alpha helix structure looks like. So you have this thing called bases in between them, which can be represented by 4 letters, A, T, G, C here. And this DNA is then used to create RNA, which is the intermediate product which you can sort of ignore or skip. But the main thing is that this DNA provides information to create proteins, which are the building blocks of life. And these are known as amino acids, which can be represented by letters like M, which stands for methionine, P, which is proline, and so on. And this is the proteins are the building blocks of life. So an analogy is that imagine if you have Lego blocks. So you can imagine DNA as the instruction manual, and the blocks or Lego blocks actually as the protein blocks, protein as the Lego blocks, where you actually do the physical things or things that actually build cells and so on. So just an interesting trivia. So the size of a genome doesn't represent the complexity of the species. So for example, for humans, we have 3.1 gigabase pairs or 3.1 billion base pairs. Here you can see a pattern between them. Mouse, for example, about 2.7 gigabases. Onion have about 5 times the size. Genome of humans doesn't mean they're more complicated than they're more complex species than humans. It's just the size of genome doesn't reflect the complexity of a species. So to the proteins, so as I mentioned, proteins are the building blocks of life, and proteins can fold and form complicated structures, which has domains, active binding sites, and so on. And these active binding sites bind to other proteins or things like molecules, ions, like calcium, and so on to form complexes. So back to the analogy of Legos. So assume that these Legos, you can form bigger Lego blocks just by combining between them or binding between them. And you can't just simply just mash these Lego blocks together. You need to build them. For example, you can put them together from top or from the bottom, but you can't put them from side to side. So these are what they mean by domains or binding sites. And protein folding prediction is actually a very big hot topic or big challenges in today's age. And a lot of machine learning is being done geared towards these protein folding predictions. So if you don't understand all that, that's fine. Just hope for you the takeaway from this previous slide is that what we know is that for a phenotype, for example, things like hearing, want to know what kind of genes involved or what are the proteins involved. And secondly, how does the protein affect this phenotype? And this is just the main takeaway. If you don't understand everything, that's fine. So now moving a bit towards my project. So convergent evolution is the acquisition of similar traits across unrelated lineages. So if you look here between this northern bluefin tuna and dusky dolphin, so one is a fish and one is a mammal, but both have evolved similar traits or similar adaptations to an aquatic lifestyle. So for example, they have a similar body shape, they have developed fins and so on. And similarly, even among mammals, you can see convergent evolution. So all these are mammals which are found across the world. All of these have a similar diet of feeding on ants, and all of them have evolved independently or separately. Adaptations for this lifestyle, they have evolved long snouts with long sticky tongues, as well as long digging claws for their particular diet. So convergent phenotypic evolution can happen through convergent molecular evolution. So what it means is, for example, for this particular gene known as prestin, which is a gene we found to be known to be involved in high frequency hearing. So if you look at this mammalian phylogenetic tree, or this tree which is built from the genomes of mammals, these are some bats, for example. So what do these bats and dolphins have in common? So I hope you all know what's echolocation. So these species they hunt through echolocation, where basically acts like a sonar. They produce sounds or very rapid clicks, and they listen to these sounds again to determine to detect for any prey within the environment. So, but it's important to know that these are bat species here in circle. Shall I put in a circle? But technically, not all bats echolocate. Only bats that hunt for insects echolocate. Bats, for example, the Pteropus vampirus, or these things, these are bats that do not echolocate. These bats typically feed on fruits, on nectar, and they don't need echolocation for their lifestyle. So, when we look particularly at this prestin gene, so the tree on the left is the one built from the mammalian phylogenetic tree, which is built from genomes of all these mammals. And the one on the right here is, if you look only on this particular gene tree, I found that all the echolocating species, including dolphin, are grouped with the echolocating bats. And this is very interesting. And why does this happen? So if you look directly into the protein sequence of this species, again within the bats, you found that there are, we found that there are similar substitutions or changes between that's observed only between the echolocating species of this and the dolphin as well. So if, for example, the non-echolocating species do not have these changes, which are found across other mammals, for example, here change from an N to a T, or change from S to an A, only between the echolocating species. At these numbers represent the positions of these amino acids. So this is very interesting because we see that the non-echolocating species do not have this change, only between the bats that echolocate as well as the dolphin. So of course, so we show that the same protein, but we change a certain key sites, might be able to affect a change in the phenotype or the activity of the protein. And of course, once this study came out as a typical bandwagon, lots of people jumped on this. This looks very interesting. So people then jumped on looking between bats and echolocating bats and dolphins, and they found more than 200 genes linked to hearing and vision. A lot of these genes were tested to show roles in hearing as well. So it doesn't mean that all these genes might play a role in developing the echolocation ability of bats or dolphins, but it could indicate that what kind of gene changes might play a role in terms of creating a change on protein activity. So what we want to do is, previously we have a known convergent phenotype like echolocation. Then people looked at candidate genes or the genes involved in hearing because that's important for echolocation. And then they found, okay, this gene like prestin, for example, could be important for this phenotype. But in this genomic era, where we have so much genome information on genes from so many species, what we want to do is we want to look at all these genes from different species, we want to predict, look for convergent gene evolution, and perhaps we can uncover a convergent phenotype. No, not just echolocation. Maybe there's a phenotype between, let's say, bats and horses, or humans or cows, for example. Then we can uncover new phenotypes or new gene associations. So the goal of my project is to find signatures of convergent gene evolution for any pairs of species, genome-wide, and also to link these genomic changes to phenotypic differences. So this is the overall methodology of the pipeline. I will walk you through each step. So first, we want to get sets of autologous protein sequences from Ensembl and align them. So what are autologs? So autologs, can we imagine if you have a gene ancestor species, and then once speciation happens, then you have species 1, species 2, like mouse and rabbit. And so autologs basically are genes that are related through a speciation event. So we can retrieve these autolog sequences from Ensembl. Ensembl is a public genome database that houses many vertebrate genomes, especially for mammals, including humans, mouse, and so on. So this, for example, this is the site for the mouse genome. There are lots of information you can play around with. But here we look at talking about autologs. So for example, for this particular gene, SLC26A5, and these are autologs from different mammalian species here, like ostrich, sorry, ostrich not as mammal, but bird, but mouse, alpaca, and so on. So that once we get these protein sequences from this Ensembl, we want to align them. And by aligning them, what it means we want to find a way to arrange these sequences to identify similar regions. So why do we want to do that is because we want to identify highly conservative or identical sites because these sites usually imply importance, functional importance, because if any changes happen due to mutation or errors in copying, and this could be little, and therefore any changes that are little typically can not to get passed on to future generations. So the site is highly conserved, usually means that the site is very important towards the protein function. So therefore, and if there's a lot of changes that happens, for example, this non-conservative site, it means that changes at this site, but maybe does not affect the protein function, so it doesn't matter whether it changes or not. So therefore, a combination of information from the conservative and non-conservative sites allows us to establish, for example, the relationship between human, chimp, mouse, rat. This just for a few species here. So the trees I showed you earlier, like the phylogenetic tree, they are built from information for hundreds of genes that have this kind of information. So that from there can imply, how do these species, or how do these species evolve. So the next two steps are sort of combined together, what we look for, which is likely to look for branches that show the same substitutions, and secondly, on how to assess the significance of these changes. And I'll sort of simplify that for the purpose of this talk. So if we go back to the pristine example, which I showed to you earlier, so we're going to look exactly for these same changes between species. First of all, here we see a change between the bats and the dolphins. So this is a very interesting change that we want to keep track of. So the pipeline that I developed as a summary, we download the data from Ensemble, lots of data, and then we look for species pairs, and we applied our approach to look for these species pairs I showed earlier across 31 mammals and over 14,000 proteins. And we came out with this of more than 1 million species pairs with at least one substitution. So why do you have such a high number of over 1 million? So again, to press back to the example. So assuming here is a what if example, the one that blocks is a representation, for example. So we see a lot of changes on this site, right? And we see, for example, a change between, let's say, between a human and a cow. It looks the same. Oh, there's a change right here. Then there's a change between these two bat species as well. Okay, another change here. So we see lots of changes here. But we look at this site, right? This site seems to be a lot of changes happening at this site. It could mean that this site is not very important. And even if you observe changes here, doesn't mean that it might affect protein function. So, and that's why, because my pipeline cuts all these small changes between different species pairs, that's why it's such a high number of results or high number of initial data. So then we applied some filtering and some statistical checks, which I will not talk about, but basically we sort of, to remove these non-conserved substitutions, so basically we remove from over 1 million species pairs, we narrow it down to over 500 genes, which we believe that happen only in conserved sites. So the next step is a quality check and pipeline. I want to just look back on the bat and tooth or the dolphin family, whether we can recover the same results. So from the hero-inherited genes, we managed to recover pristine as a control. So okay, good, we can recover what's already mentioned in the literature. But additionally, we also found some additional genes related to hearing, which has not been reported before. And we found that these genes have been reported where mutations in these genes could lead to hearing loss. So that's pretty cool. But more importantly, we also found a few genes involved in fast skeletal muscle, 3 genes here, myosin heavy chain 2, SERCA1, and also casequestrin 1. And these skeletal muscles means the muscles are attached by tendons to the bones. That's what it means by skeletal muscles. So, so I'll just talk about the 2 genes highlighted in red here for the purpose of this talk. So for this first gene, the change from E to a K at this position is highly specific to only 2 aquatic mammals. So this is from for this particular gene. So the megabat and black flying fox are bats which do not equal okay. They are large bats which feed on fruit and nectar. And we see a change from an E to a K only between echolocate species, the dolphin family, and the echolocate bats, and not in the non-echolocate bats and also in other mammals as well. And this is a very highly conserved site. The same thing for CASQ1. Again, we found a few sites that observed only between the echolocate species, and yet the non-echolocate species like megabats and humans and so on do not have this change. These 3 sites in particular are very interesting because you look at the protein structure. For example, there's a representation of the proteins. I found that the changes here are very close to the calcium binding sites or calcium ion binding sites of this protein. So it's very close to the critical sites of where the protein works. So what does muscle physiology have to do with echolocation? So there are two components to echolocation, right? So one is to hear the high frequency sounds, because you need to hear the sounds again, but you also need to generate those sounds in the first place. So you need to produce these clicks or these rapid clicks and pulses before you can hear them. So here I'll just play a short video of an echolocation of a bat catching prey. So just listen to the pulse of the bat. So much you can see in the middle actually is the light. The white light is actually the insect itself, and then you can see the bat diving towards the insect, and you hear the rapid change of the pulse as the bat closes on the prey. I'll play it again. So if you notice, there's two stages or two kinds of sound the bat produces. So first is the approach phase, where it sort of produces sound in a very consistent manner. It's going to approach phase where just sort of looking around for prey. And then once it finds a prey, it goes in the buzz phase where it produces really rapid clicks at the last part, because it wants to know the exact location of the prey. Secondly, the prey makes any sudden moves, you can anticipate that move and adjust to it. That's why you can see this rapid production or rapid sounds at the last part. This same technique is also being used by dolphins as well, the dolphin clicks, where they as they zoom in on their prey, they increase the sound production to get the exact information of where the prey is. So first of all, you can appreciate that both these lineages, like the bats and dolphins, evolve echolocation, and secondly, they also evolve the same technique of how they apply this echolocation. They're having this very fast production of sound at the last part. So this voice or these sounds, right, they're produced in our throats, or vibration produced from our muscles in our throat and larynx. So in bats, these are termed as super fast muscles because they can contract up to 200 times per second, which is not very common for skeletal muscles, especially among the fastest in the mammalian kingdom. So I can't go through in detail of how the muscle contraction cycle works, but basically just to takeaway is that there's two stages in the muscle contraction cycle. One is the contraction period, and one is the relaxation period. So when the nerve stimulates the muscle releases calcium, and then when the body pumps the calcium out of the muscle relaxes. So essentially, both these stages are controlled through the release and binding of calcium ions. And this is the zoom in of a muscle fiber. Again, you don't have to understand everything, but basically, as I mentioned, the contraction cycle and the relaxation cycle of the muscle. And Cas1 is the protein in dark green here, where it binds calcium during the relaxation stage, and it releases calcium during the contraction stage. So therefore, back to the question, what does muscle physiology have to do with echolocation? So a hypothesis is that the convergent changes in Cas1 or these following genes are involved in the adaptation of super fast muscles in echolocating mammals. Basically, change in one gene might not be sufficient to change in phenotype, but changes across several genes that work together might contribute towards the change in this particular phenotype, in this case, the production of sounds. So the last part here is some experimental validation. I'll just talk only about one part of it. So this experiment we did for a collaborator in the US with Jun-Hoe Lee. We did a light scattering assay. So in essence, basically, is for example, you have a solution of clear water. If you put a drop of red ink, then the whole thing becomes cloudy with red, right? So a similar concept here. So once Cas1 binds calcium, it forms complexes which turns cloudy. So here we measure the concentration of Cas1 in the solution of calcium ions. I found that the Cas1 from dolphin and bats, for example, could bind calcium at a much lower volume, at a much faster rate as well compared to the mouse Cas1. So even the same protein, but different activity, slightly different activity here. So perhaps this could be attributed to those particular changes in these amino acids, which I mentioned earlier. Of course, we need more further studies to show any, to verify this, but that's actually the extent of my my PhD project. But basically, in summary, is that we developed a pipeline to look for genes showing signatures of convergent evolution. And we found 3 candidate genes related to fast muscle activity in echolocating bats and dolphins. And our hypothesis is that convergent molecular evolution is likely involved in the evolution of superfast muscles in echolocating mammals, and although, of course, more experiments are necessary. So just stepping back and taking a wider outlook, why do we do this basic research? What can we learn from them is that, sorry. So I know my colleagues, for example, use a similar approach where they look at bacteria. So for example, can we determine like what are the changes that turn the bacteria, for example, from benign towards versus disease causing? Like we have E. coli, which is lots of bacterial strains. So only some strains infect humans and cause disease, while the rest do not cause disease. So are there particular changes that turns a strain from benign to disease causing? Or similarly as well for virus, there's other certain changes in the protein that can more likely change the virus activity. And thirdly, for example, in terms of gene therapy, you can understand, for example, find which genes are related to are targeted by that phenotype or the particular disease, and then what are the critical sites in the protein. So as acknowledgement, this is my group. I did my PhD at MBIOS CBG. My PI or my group leader is Michael Hiller, is the fourth on the left here. These are the institutes involved, and that's the wrap up from my PhD project. And now I'll shift the gears a little bit. I'll talk about what I'm doing currently. So I'm working as mentioned earlier, at the Quantitative Biology Centre, or Tuebingen. In Tuebingen, this is the view from I think it was from the hill. I think the building, this is the one I'm looking at. It's a bit grim. It's I think just a couple of days ago. Here am I at my desk, and yeah, nothing fancy here, just me and my computer. So I'll talk a little bit about what the institute does. So Tuebingen is a sort of a facility or intermediate between the client and sample processing facilities. So it's within the Institute of Tuebingen, which was created with the funding from the Excellence Initiative and the German research funding. So basically, we are as an intermediate between the client and the other facilities in the university. So most of our clients tend to be from the university hospital, but although we have some corporate clients as well, or others from neighboring institutes or neighboring universities. So for example, they bring in some samples, we provide them things like consulting, experimental design, sample handling, like they have tons of like DNA samples or whatever, RNA samples. Then we do a barcode registration and data storage. And then we have, we do analysis for them as well. So basically, we can talk to a client of, for example, they want to run an experiment, they have tons of DNA samples, want to design experiments, to provide guidance to them. And then we give us like the barcode and so on. Then the client will deliver the samples to these facilities for sequencing, proteomics, or protein analysis. And then they will give the results to us. We store the data, we analyze the data, and then we provide the results back to the client. So there are three departments or three groups within the Tuebingen, which is the RDDS, Research and Development, the ITSS, IT Infrastructure and Scientific Software, and the BioPM group or Project Management group. So the first department, the first group is they work more R&D, as the name says. They work on developing new pipelines, they work on new methods of analysis and so on. The second group, they work on developing software, like things like data storage, handling data storage, how to register samples, cloud computing, or the cluster servers, and so on. And actually, I'm in the third group here, project management bioinformatics, where we, as I mentioned, we do a lot of project management. We do client meetings, we produce offers for the clients, we execute the pipelines, and we prepare reports. So let me shift back a little bit and tell you about, introduce the era of big data, which is what I titled my talk at. So right now we live in the era of big data, and I think you probably heard the term a few times. So what do we mean by big data? Is that it's characterized by the size, complexity, and the rapid pace of combination of this big data. Data is coming in in large manner in different forms and a very fast pace as well. So here's a graph that shows some of the DNA sequencing cost over time. So in 2003, the first human genome was first sequenced and cost over 10 million. And then the cost continued to drop. 2005, there's new methods in sequencing. In 2014, the cancer sequence are much cheaper, about $1,000. And today, of course, you have hundreds of genome. I mean, the tech is much faster. You can sequence the genome within a couple of days. And even like companies like 23andMe and so on, they can sequence the genome over like a week or so on. So the cost has dropped a lot. And that's why we have this rapid accumulation of data. And this is why one of the big aspects is machine learning, because you need a way to process this ever-growing size of data. So the next stage, at least in the pharmacy healthcare, is in Germany. What they are pushing here is at least this thing called personalized medicine, and this is technically in German, but I think it's quite easy to understand. So basically, a change in our current approach is there is a one-size-fits-all versus and want to move towards more individualized approach. Yeah. So I've translated the ones at the bottom here. The 3 labels actually means like strong side effects, low effectiveness, and strong effectiveness. So basically, the one size fits all. You provide same kind of treatment or same medicine to everyone here, and then you have this level of results from strong side effects all the way to strong effectiveness. So we want to move towards an individualized approach where we can determine, for example, predict what kind of possible reaction you have to a particular treatment, and then we assign the treatment that best suits you, the best suits you. So the treatment that hopefully have the lowest side effects and the maximum effectiveness. And this personalized approach, I think, is a lot of investments or a lot of research coming into this area. And one of these big or one of the most things I would say approach coming up is this new technology known as single cell RNA sequencing, which is I think it's being used a lot by the university hospital and so the groups around here. So as introduction, so for example, in your body, there's various cell types in tissues, right? You have red blood cells, immune cells, and so on. And when you talk about cancer or tumor, it's important to know what are the exact cell types within the microenvironment of the cells. Basically, for example, here's the liver. You have all these different cells from the liver, different parts of the liver. And you need to know like the microenvironment. So basically around each cell, they have a slightly different environment as well. So currently, when you take a biopsy, for example, you take a slice of tissue, you take hundreds of different cells within that small sample of tissue. You can't distinguish between different cell types. So what we want to do is that single cell, you want to isolate single cells and want to analyze the difference between healthy versus tumor cells. And a lot of these studies are being done right now in liver tissues and skin cancer. So here's an overview of single cell RNA sequencing. So as mentioned here, for example, we take a tissue, a tumor tissue. So now we have technology to sort of isolate and sequence individual cells. You sort of can tag each cells individually, and then you run the analysis here. You can sequence the RNA for each cell, take a read count, and you can find the gene expressions. Then you can sort of even classify them according to different cell types, and then you can sort of analyze them like this particular cell, for example, here. Like what's the difference between macrophages and the cholangiocytes? How the gene expression differs between them. So previously, you take one sample or a tissue sample, you cannot distinguish between cell types. You have this gene expression, but not sure exactly where the gene expression comes from, which kind of cell types. And I think this is one of the biggest or most popular approach right now. I think I have like 4 single cell projects from different groups right now relating to skin cancer and liver cancer. So as of as winding down a summary between like what am I doing right now and what I did previously. So in a way, comparing between academia and my current role. So I'll say that previously I had one huge PhD project, as you saw earlier, that took me like 4 years to do the pipeline to look for convergent evolution. So right now I have actually several projects running at the same time. So I guess there's a difference between one main project and now I have several projects at the same time. The duration of project so differs. Like the previous one, it took me, I guess, 4 years to do my PhD. Right now, I think they're relatively short. They can be between a couple of weeks or over months, or depending on the complexity of project. And here, instead of working from scratch, like the previous I developed project all the way from getting the data, processing it, pipeline, and then validation. So here, usually we step in at just one part of the project. So basically, we have the university hospitals with the blood samples or DNA samples and so on. Then the facility process them. Then they give us the results, and then we just step in at the data analysis part. Now we analyze the sequence, we analyze the DNA data or the RNA data, and we give the results to the client. So the types of project right now, as I'm in the bio-PM group, I'm less on the method development. I don't really work on sort of developing method from scratch anymore. Usually we use the pipelines that are already available or develop or tweak what are available from the RDDS group. And also because previously the type of project is that you compare my PhD project, it's at the very basic level. We're not sure what we're looking for. We try to have a new approach and so on. But here for my current role, we have human and patient data. Also, it's more applied in terms of cancer cancer research. And of course, when we talk about human and patient data, we have to be more careful. There's like ethics evolve into like patient anonymity and so on. So we need to be how to handle data is a bit different as well. This is a group photo that we have with the link to our website, and this is I think the recent retreat that we have a couple of months ago. I think south of Tuebingen, I think was near Stuttgart. Yep. As the final slide outlook, so I think I think probably my journey is a bit different than most people. So I started off with my degree. I started with more like wet lab. We learned about biotech and molecular biology. So I did like PCR, DNA sequencing, RNA stuff, RNA sequencing, RNA extraction as well. Then I got interested in phylogenetics, but I got interested more in bioinformatics, basically more computational aspect. So that's why I think from master, I did about 50% in bioinformatics. And then my PhD, I sort of moved full on towards computational biology now. And I think for my role, at least, I think I'm trying to move a little bit more from academia or basic research towards, I guess, hopefully to move towards industry. So technically, I'm still working at a university right now, although my role technically is not really, I'll say, academia anymore. It's sort of, in a way, half-half or mixed between industry, because you have different projects at the same time. You sort of work towards a particular goal, and you don't just keep trying again and again to solve the problem over a long period, like over years. And I guess I'm trying to expand my skill set, not just focus on phylogenomics, which is very cool to investigate evolution relationships, but technically it's probably not very something that's in demand industry to examine evolution of species. So things like RNA sequencing, transcriptomics, things are like involved in the medical aspect, things are much more applicable, and I think I want to leverage that towards a more generalized approach using data science and more machine learning, which is as I mentioned, getting more popular these days with dealing with biological data. And with that, that's all. And thanks for listening to my talk, and thanks for attention. And I'm happy to answer any questions that you have. Thanks.
Thanks, Dr. Jun Hoe. Thanks, Jun Hoe. Your experience and journey is quite interesting, and it's quite complicated with all the knowledge, everything. So it's just we have some questions in Q&A. Yeah, I want to ask. Inside a web lab, would it be beneficial to know coding language like Python's? Like, do you think it is something that students right now can learn online? Like, how do you think is it like quite difficult for students to learn this, or is it essential skill sets to get involved in bioinformatics field?
Yeah, these days it's really hard to be like a pure wet lab anymore these days. Even if you get some data, so you need to analyze, at least you can't just you need to know at least some basic level, because you need to deal a lot of data these days, like genomes and so on. I think it's very hard to just be a pure person working at the bench anymore. Sure, yeah, I think Python is good. Good start. I think one of the most simple languages started Python as well. I think it's probably the most widely used computer language as well, especially for biology. I think the next option is R, but between Python and R, I think Python is a bit more accessible. And I think it's just good to learn some basics as well. But even if you don't learn Python, I think you need to familiarize yourself with certain tools which are very commonly used, things like NCBI, BLAST, I think Ensemble, I think things which are very commonly used. So you need to probably be familiar with them if you don't want to learn programming languages. But yeah, I think it's good to start.
Yeah, and do you reckon about except from Python's, any other coding language that that's right now the industry are looking for, what kind of language they normally use, or it doesn't matter really much?
Yes, I think Python is a good start. I think next is R. R is also very popular, but I would say if you have not started before, I think Python is probably easier to digest. R is a bit more challenging, and I think it's a very good support, but it's more complicated and it's a bit more narrow. I think whatever stuff they develop in R, usually they will develop stuff in Python as well. But yeah, and so not just this language, but also working with the terminal, like working with the command line. I think there's also some skill that you need to know as well.
Yeah, totally agree. Yeah, and there's another questions how did you find the topic for your PhD's research? How did that connect to you? How do you get involved in that research topics?
So actually, the topic was proposed by my PI or my supervisor at that time. So I was just looking for a PhD position, and I saw this. He was starting a new group in MPI CBG in Dresden. So I just wrote to him saying that I'm interested in kind of what you do. And actually, I was a bit worried because, as I mentioned at the time, I was still not computational yet. I was still 50% doing wet lab, and my computation skills are not super amazing. And he was my PI was a bioinformatician. So it took me a while, but then he was interested in what I did previously as well. Then we sort of got talking, and then he proposed this topic, which I was really interested in, and then that's how I got started in it.
Yeah, this is quite interesting. Like, it's just like a small push from others, and then you got involved in like changing a whole difference and also about the journey itself, right? I saw that you did research in like sea bass, microbes, flatworms, like phylogenetics works on those. Is it hard for like changing? Is it hard for like to relearn the skill, or is there any difficulties you face in changing like the species to get involved in get the research involved in new species?
I think that depends actually. So between mammals and flatworms is still not too bad because they're still, I guess, eukaryotes, or I guess animalia. But from mammals to bacteria, that's a bit difficult actually. I think I took a long time took longer to understand the differences between them, because for bacteria they don't have we call it, I'm not sure you notice the term I use, metagenome, not genomes. Because the idea is that you take a sample of soil, for example, right? You have hundreds of bacteria or virus in that particular small drop of sample. So you don't actually know which bacterial strain, which genome sample come from. Compared to, for example, like human, you take one drop of blood, it comes from that particular human, right, or particular species. So there's a challenge or difference in how you sample the genomes for bacteria. So I think in that case, there was a bit of took me a little bit longer to sort of figure out the difference between them. But I think what you need to know is at least some basic computational skills, and then I think it's always a learning process as you go along. You can't just keep evolving.
Yeah, agree. And I think quite a lot of our audience are interested in like how you get your journey in outside Malaysia. So there's some questions asked that, seeing your journey, you have tried a lot. Is there any advice you can give for fresh graduate in figuring what they want to further study in master or PhD?
You mean what do you like when they apply overseas, or what do you mean exactly?
Oh yeah, actually, this question is asking about, is any advice that you can give to because some of audience over here are figuring about to study in master and PhD. Do you have any advice for them?
I guess it just depends on what kind of what's your interest. I think that's the main thing. You need to figure out what you like to do, what you don't like to do. Like I think towards my end of my final year project, and also my doing master, I realized I like to do more computational. I think I kind of not that good at wet lab anymore, because I know I was getting tired of doing like tons of PCR, and they're so tedious, and I was like too lazy to extract tissues anymore from seabase. So I was just towards computational stuff, and then I was just looking around, and then I tried to apply. As for moving overseas, I think, I just need to sort of adapt to a different lifestyle. I think sometimes it's not easy when you move to a different country, but yeah, once you get used to it, you keep an open mind. I think that's how you go about it. Yeah.
Is there any like language barrier when you went to German? Like how do you overcome that language barrier if you have?
Yeah, I think you just need to keep an open mind, and I guess motivate yourself. Sometimes it's quite challenging in terms of language. Like when you came to Germany, at least it's I mean, at least most places you apply to or you work in, usually there's like in the university, usually speak English is fine, so as the official like working language is fine. But if you step outside the university and you go about daily life, there's lots of it's very hard if you don't know German at all, because like you go to the shop, even the banking system is always complicated for me. Everything's in German. So yeah, there's some language things you need to adjust to get used to it. But yeah, but so if you can take your pick, like I think Germany is a bit hard. France is probably hard as well. Sweden and Netherlands is they speak perfect English. Everything. I mean, they usually have documentation in English as well, and they speak English very well. So that's probably easy in terms of language barrier. But yeah, just something just depends on your interest and your work. If you like it, then you sort of just get used to it.
Yeah, I think also for Malaysians, like we are multi-language, like it's quite easy for us to pick up. Like it's just whether we have the chance to practice it, and then we have that mindset, to open mind to embrace the language itself. Yeah, and for a lot of audience also want to know, is there like any opportunity for Malaysian students, like bioinformatic Malaysian students in Malaysia? And do you think that is it is it easy for them to get a career in Malaysia, or is it better to outside Malaysia?
Yeah, unfortunately, I don't think I can help much because I don't think I've been in Malaysia that long anymore ever since I did my PhD, so I can't really comment too much on how things are in Malaysia. I mean, I still remember during my first, when started my degree, actually, they just introduced the new program, a bioinformatics program in UKM, and they asked us who was interested to join the bioinformatics program. And I was like, I don't know what it is, and I just stick with my zoology. So yeah, I can't really say for certain how good or what developments of bioinformatics in Malaysia is. But yeah, at least here in Europe, I see, or in Germany at least, a lot of countries here, I think bioinformatics is quite a big aspect. A lot of them have a separate bioinformatics facilities of a bioinformatics group, which do a lot of this, basic bioinformatics analysis. So I think probably it's a bit more, I guess, more wider, or there's more opportunities probably in terms of bioinformatics. But again, yeah, depending on what kind of areas, even bioinformatics is a very wide area by itself. There's things like, you're doing transcriptomics, genomics, like, or phylogenomics as I did previously. So a lot of things which you can sort of go deeper in.
Yeah. How about for students that are, I mean, for the opportunity in Europe sites, like, do you reckon like Germans is the best if bioinformatics students want to and continue their study or continue the works over there? Or do you reckon that there's other country that they have more opportunity in this field?
Well, I can speak from experience from the countries I've been in. I think, for example, like Uppsala University, the one I did my postdoc in Sweden, I think they're very good bioinformatics program. There's lots of good groups there as well. So you can look up, I think, SciLife. I think that's one of the scientific hub between a few countries. So between Germany and Sweden as well. So SciLife, I think, is a big bioinformatics program which people interested can check it out and see. My university right now, Tuebingen, also has some good bioinformatics stuff. You can check out University of Heidelberg. There's also very big research facilities, lots of bioinformatics. EMBL, yeah, so I think lots of opportunities for bioinformatics. Even the MPI, the place where I did my PhD, MPI CPG, they have a PhD program there which I enrolled in. So they have 2 intakes twice a year. So I think once is the winter and one's the spring intake, one's the fall intake. So you can even apply that as well. And yeah, it's a very good prestigious program, full stipend and everything for about 4 years. And so yeah, if you can get in, that's a really good opportunity.
Thank you, Dr. Jun Hoe, for sharing this with us. And also another question, what is the trend now in the phylogenetic fields? Like, I think because of COVID's virus research, looking in the phylogenetic trees of virus is one of the big projects. Is it another trend that we don't know? Like any specials that you think is interest to mention?
So I haven't done phylogenetics that much these days anymore, but at least from what I know, it's like the metagenomes, the macros, for example. I think there's a big push now towards metagenomics due to, as I mentioned, machine learning as well. So they're trying to, like, as mentioned, right, I call it metagenomes because you have one sample, you have hundreds of bacteria or virus, whatever in one sample. So how to separate them efficiently? So machine learning is being done a lot for that. And I thought because of the climate change, so like, I think like some studies, for example, doing metagenomes of how for example, from deep in the permafrost, they're being released now. How are these changes in this genomes of this bacteria or virus from these particular areas, like climate change, the changes of this pollution and so on. So I think metagenomes of bacteria are this sort of growing, especially with the changing environment, at least. And from us, what I'm doing right now, I think, the medical data or medical informatics, I think there's a big thing that, like I mentioned, the push for personalized medicine, so the RNA single cell sequencing, and even right now I'm doing like spatial transcriptomics. It's not just single cell, but where the cell is located as well. Quite spatial RNA-seq. Yeah, so basically, like, not just which cell came from, but where exactly the cell, the front or the back of the tissue and so on. So in terms of space. That's the next thing I think I see people are pushing there now in terms of RNA sequencing.
All right. This is I'm getting more interest in the topics. For me, right, I'm actually doing in marine biologies, and I'm right now doing on. I have colleagues that are doing in metagenomics of seaweeds, so I understand a bit of what's the term that you're using, the words, the terms that you use. Just another question is that the question is, how do you think, like except from the coding language, everything's right? Do you think understand about statistics, like PCA, HCAs, those skill sets are important in bioinformatics?
Yeah, I say again, it depends on which aspect. I mean, for me, it's always impossible to learn everything there is to learn.
Yeah.
I mean, mathematics, I mean, there's too much to learn. Like, at times I also feel that I wouldn't call myself as a bioinformatician because I don't think I came from a computing background. My background is in zoology. Actually, the reason I got a PhD in biology is because I think when I was supposed to sign up for the university, I couldn't get enroll in the informatics department or the PhD in informatics is because I say my background is in biology. I need to have 50% background in IT, then I only can join the faculty of informatics to get a degree there. That's why in the end I end up with a PhD in biology, even though technically I didn't do any lab work, or I just do mostly on computational. So yeah, I think it depends on what you want to do. If you want to do more machine learning, as I'm trying to do now, actually, I'm trying to learn more machine learning. You need to probably know more about some of PCA, something about calculus, and something about algebra. If you want to go towards more medical, then probably HLA typing, those kind of things, depending on which area you're going. Yeah. So yeah, there's just too much to know. I think you need to sort of figure out or try to learn along what you need for your area.
So basically, like have to have a keep on learning, and then the world just keep changing. So you just have to be open mind with the skill sets, the techniques that are coming in. Yeah, I think there's a lot of questions. There's questions here in the chat box. Is that I think in the background as well, there's a lot of people keep asking curious about different language, because like just now mentioned about there's Pythons, there are R, there's C++. A lot of people are asking about which, I think they're quite curious about this question, which are the most easiest to learn, except of like in your personal view, like
I think
which one?
Yeah, I think Python is the easiest. I think it's the one that is the most English the most, so it's more logical, and I think Python is always a good start. I recommend Python. And then if you want to go deeper, I think R probably. But I think these are the two most frequently used ones. Unless you want to go really specialized and you want to develop software, then you need to learn like Java and stuff, or HTML. But that's a bit more, I mean, if you're more like a developer, programmer, which I'm not actually, so I just mostly do the analysis, so I just mostly stick with Python.
Thank you, Dr. Jun Hoe. Yeah, I have questions. Another questions over here is, this thing is from academy. Going through the research journey, it could be lonely and lack of motivations. Like for you personally, like what are the things that keep you motivated, keep going in your PhD study, and also like right now when it works, like definitely feel, especially like right now the COVID era, especially sometimes you feel demotivated. Like what do you do to keep yourself motivated?
Yeah, I think it varies depending on your how I guess your personality or something. So for me, I like what I'm doing. I like living the experience here. So I guess it's fine for me. And yeah, I guess if you want to read on a personal level, I think it's a bit harder, especially during special occasions. Like for example, I don't think I've been home for Chinese New Year for many years already because the timing is just not right for me. Like I did take my holidays during Christmas. I travel back about once a year during Christmas, not because I celebrate Christmas, but because it's the most convenient time since everyone's gone. The whole place is sort of closed down, so it's easy to do holidays then. But then that's fine for me actually. So yeah, it really depends on your personality or what kind of things you like. And for me, I like working here. I like the environment here as well. So for me, it's not a big deal for me. And yeah, but I guess you sort of know or how to expect, like, things like, for example, like different holidays, celebrations, you might, want to adjust for these kind of things.
Yeah, thank you. Thank you, Dr. Jun Hoe. I think, let me just check with the Q&A sections in Facebook. Oh, there's a question over here. In your opinion, do you recommend postdoc for PhD graduates to improve their skill set before entering industry?
Yeah, probably no. I think if you can't apply to industry, then you definitely just go straight on. I think actually a lot of people nowadays, I think it's a trend at least. And they realize that academia is really like a tough world out there. It's really hard to be I think like 1% or 5% to secure your way towards getting a grant or starting your own group and so on. It's very challenging. I've seen a lot of people who didn't quite make it. Like a lot of talented people. I think when I was in, I know of like, I think one colleague in Netherlands, for example. I think he was doing a postdoc and then he finally managed to get a grant after 10 years. Which is great for him, but imagine staying being a postdoc for 10 years. It's a very strong, long commitment, and you're not sure whether in the end you get it, but he got it in the end, so it's really good for him. But still, it's a very tough challenge. So, yeah, I think these days there's a trend of appreciation of moving towards industry. It's not a bad thing, you just like any other thing. You finish your degree, you want to get a job. So if you want to, you can start applying even after a PhD already. In my case, for example, because I was not certain what I want to do at the time, so I thought, okay, I just do a postdoc and see how it goes. If I can make it, then it's great. If I not, then I'll see how it goes. So I guess I didn't quite make it in terms of, I find out that maybe the terms of commitment, as people like, they have very good ideas. They have this big long-term ideas of what you want to do in terms of research. They want to have different aspects. Like my boss, for example, I'm doing a PhD. So my aspect is just one aspect of his genomics big ideas. So I was doing convergent evolution. I have other colleagues who do, for example, on gene losses. Another colleague doing on evolution of different limbs, like species that lost their limbs, for example. So they have this big idea of different how different aspects can come together. And I don't think I'm the kind of person who has that big ideas. Like for me, I like to do research, but I don't like to like spend a lot of time writing grants and planning this whole big research network or this big idea. So it really depends on what you like. And yeah, industry might be a good thing to do. And I think sometimes people get frustrated as well. Like, if you see from things like, I do some basic research on evolution and then it's so what? People don't really care, or you might want to do something that's more tangible in terms of medical or things that you want to feel more useful. So yeah. So if you can start applying, after a master's, after a PhD, you don't have to continue on a postdoc unless you really like the area a lot. Like some people I know, they have some options. I think some companies, they do offer like a corporate sponsor postdoc. So I think like is it Merck or Pfizer? I'm not sure Pfizer, but some companies do offer like, they sort of you're sponsored by the company, but you do a postdoc in university, and then after 2 years, you sort of your research, whatever sort of helps, is sponsored by the company as well. And so then in the ways of training, like like between the company and the university, you can do a postdoc. I think some of my friends do that as well, actually. So if you find something like that might work for you.
Yeah, I think different personalities and different people have different preference. Some personalities or some people prefer in academia, some prefer in industry. Yeah. One question is that I forgot to ask this previously. How did you get into your current position in Q? I mean, how do you get into QBIC?
Quebec, yeah. So
yeah.
Quebec, yeah. So I was, as I mentioned, I was in Dresden. I was doing my second postdoc in sort of wrap-up postdoc. So not wrap-up, but second short postdoc in Dresden. And then COVID happened, and my group leader at the time was quite kind enough to extend it for a few months because I was stuck in Germany. And then I was just looking around for different positions, and I found a position here in Quebec, so I applied for it, and yeah, that's about it. So,
yeah, it's actually because I'm actually right now in Malaysia, but previously I'm studying in Australia, and then I did also apply same situations like you. I apply in a lot of company in Australia as well, because that time I have a also start in Australia because COVID, and I got a postgraduate visa. And what happens is that for me, this is just my personal cases. Like a lot of company, they didn't want to hire because of their fear of the COVID the situations. And slowly I end up found are open up for graduates, PhD students. So I was like, since I can't find jobs over Australia and other places, and right now just apply for it. And then and then I think just history. Right now I'm just starting my first semester in PhD. And then here lead to my next questions. Somebody asked, like, is there a way to speed up a PhD? I want to take a PhD, but I'm scared it will take very long. Any wisdom for him?
Yeah, I guess depending. Yeah, but I think you need to think. First of all, you need to think whether why you want to do a PhD in the first place. Like it takes a long time it takes a lot of commitment actually to do a PhD. It's not an easy thing to do, and a lot of times you might want to, you feel like giving up. I know some people who they did their research, they were fine, everything was cool, but then the last part, the writing thesis part, they sort of give up because there's just too much, write the thesis, and it was a bit sad because you spent 3 and 4 years, but then the last part you just couldn't do it, and or some people just quit after 2 years. It's long thing to do. It's a lot of commitment. Sometimes like things, your experiments fail. I also experienced that before. My results fail, and you don't get what you want, and you're thinking, you're questioning your life choices. No, what am I doing right now? Kind of things. And I think really, probably all PhD students have one point during the studies, they're like, what am I doing here? You see your friends partying or your friends celebrating, and then you're stuck in the lab on a weekend doing these kind of things. So sometimes it takes a bit of commitment and self-motivation. And I think before you want to do a PhD, you think of why. If all you want is just a paper or title, I think it might not be worth it because of the time commitment. If you just want it for career advancement, I don't think it's actually that important for career advancement. If you want to do something, if you can get a job in the industry, experience-wise, that could be fine as well. It's just a title, but if you work in the industry, you get experience. That's nothing wrong with that as well. So if you want to just advance a career, better just stick in your job. You get a stable job. You just work in your regular hours. You get experience, and you need to think why you want to do a PhD in the first place. I don't think there's a way to fast track a PhD unless you're really good. But even then, a lot of it comes down to luck. Sometimes some people, the results just work. They got everything to do in the first run, everything works, and they could do their PhD within like 3 years.
Yeah, I think this, like you mentioned, this all depends on what's the destinations, like what's the final goals they want to come out from. Like, if they just want to for career, they can straight go for industry. But if they're looking for some like make change to the world, or they're looking for really involved in academy, I think continue PhD is one of the way to go. And we have questions over here. Somebody asked Dr. Jun Hoe about your advice for students seeking master and PhD opportunity abroad. Oh yeah, how do you get the opportunity to study in Germany?
I think you just look up the websites. I think there's probably a few. I think now this is easy to look up. There's lots of different programs. Just apply online. As I mentioned, the institute where I'm at, where I do a PhD, and CBG, they have twice a year intake for PhD students. You can try to apply there. Just look around. I think the easiest way is that it takes a lot of time, effort as well to look around, look for projects or PIs that you're interested in. Like if you're interested in phylogenetics, for example, I actually applied to a lot of places. I like the work you're doing. And then you see whether the group is hiring or not. You talk to the PI first directly, it's easier. Don't just apply directly to the program. Usually you talk to the group, discuss what kind of things you like to do, and then he sort of find out whether he likes what you've done so far. Then once you have a good rapport, then maybe you can apply for the program after that. But there's just a lot of websites. I think it's a lot of time and to find out find a group that you like, then write to them whether they're hiring or what kind of projects you can do, and then establish some connection, and then go from there.
How about like, why do you pick German? Like, do you have like, is your original target Germans, or like you just found an opportunity, just take it and then just go?
Yeah, in my case, I was applying to a few different places, including UK as well. As I said, I think because PhD is a long journey, right? You need to be really motivated, interested in it. So you find like if you get like a spot in the US, but you kind of like, you don't like the topic, you go there just for the country, but after 1 year, you probably feel like, like you kind of hate what you're doing, it's like the motivation is gone. You're just there for the place, but you really don't like what you're doing. So it's very difficult. So I think the main thing is the interest, and also the sorry, the PI or your boss. You need to at least have a good relationship with him. Like, if you kind of hate your boss hates you, you're so very bad in terms of your project as well. So good interest, good PI, and then of course I think the location for me comes third after that. So it really depends. Unless you really have a fixed idea, like for example, like I don't want to go to France, so you don't apply to France at all. But I think the main thing is you pick something that you like, and then you consider the country. At least in my advice, the research interest comes just before the location. And if you don't like the location, then I guess don't apply there in the first place. But
yeah. Mention about PI. This leads to another questions. This is about supervisor. If you apply online for overseas master or PhD, how do you know the program is good, or if the team is good? Like for supervisor, like how do you know that supervisor is good? For me personally, I normally check the this is one of my friends told me, it's a tricks that check the publications, the team, and also like how long it is the supervisor, the team is in that field of study. So if the supervisor is very is involved in the research a long time and you got publications, most likely when you apply for PhD under that supervisor, he or she will just ask one of the postdoc or one of the PhD students, final PhD students, to mentoring you, so you didn't get any involvement with that supervisor. So the trick is to find a supervisor that is just entering the career, and then they also got quite a lot of fundings and publications coming in as well. Yeah, this is just my opinions. How about Dr. Jun Hoe?
It's tricky. I think it's no one right way to do it. So yeah, I think usually you write to for example, for me, in my case, if you're interested in a project, you just write to your PI, whether they have an opening, what you'd like to do and something like that, and then might see whether they have an opening and they want to hire a PhD student, and then you can talk with him more. I think the next step actually is easier if you just talk to other students in the lab, or how's the project, what I'm thinking of applying there, like how's the how's it working in the lab, how do you do daily? I think that's what happens at least in my group as well. Like sometimes the new PhD students want to apply, they will talk to my boss first, and then after they will talk to all the people in the lab as well to find out what the group is doing and how the daily life is and so on, and how the recommendation is. I think that really works. If you like the idea of like very hands-on supervision, then you can find out what kind of style your PI is based on the people working in the lab, whether he's a hands-off or hands-on thing. In my case, it's actually very tricky. My PhD in Germany, my boss actually he was just fresh off. I mean, as in like he just finished his postdoc, he just got a position in Max Planck, and was looking for people to fill up his lab. So I was the first person to join the lab. So I was there before any postdocs or anyone else was in the lab, and I was there for a few months before even the next student came along, and the next postdoc came along, and student came along. So it's actually very which was nice, I guess. Yes, but also very stressful because, as I mentioned, he was a bioinformatician and I was not. I was still halfway through my polishing my application skills. So all his focus is just on me alone. That was quite quite stressful actually for the first few months. So and I guess he was also new, and he's still learning how to be a PI. So that's good and bad in both sides. I won't say that it's good to join a fresh PI or it's good to join an established PI. As I mentioned, if an established PI, I know all my friends, his PI is one of directors of the institute. So as a director of the institute, he has lots of responsibilities, and there's a huge lab with lots of postdocs as well. So I think he meets with his PI once every 6 months. So yeah, so then most of the time it's done by postdocs, and yeah, so I think there's no easy way. You just talk to the people in the lab, find out what's the experience, and then you see whether that kind of management style fits with what you like.
Yeah, I think this is like some of the tricks, some of the way we can know like whether that environment is good for you, and that expected as well. And here's a questions like, this audience asks about like if I graduate with biotech degrees, don't have much knowledge in computer science, where should I start if I like to go into data science? Is it straight start in the language but if for bioinformatics, it starts like straight start in like for self-learning, straight start with the language, or get like knowledge in some specific courses?
Yeah, I'm not sure I'm the right person to ask this, because technically I'm still learning a lot how it goes along. So I'm not sure that I'm expert in this. I would say that, yeah, a biotech degree is probably, if I remember correctly, or how it is in Malaysia, or at least in UK, it's a very general degree. So yeah, probably there's lots of online courses these days. You can sort of brush up your skills, as I mentioned, some basic skills, like, if you want to move towards data science, I think Python is probably one of the easiest, or one of the critical skills that you should know some things about. And then you can take up lots of courses like mathematics beginners and so on, things like, sequence alignment, as I mentioned earlier, using NCBI, using Ensemble or Galaxy. These are very common platforms, which I think people might expect you to know. So yeah, just look up, there's tons of articles or online courses, just few things to get you started. But if you want to go deeper, I think it really depends on what kind of job that you're doing. Like some people, they like protein structure prediction, so they study a lot just on the protein modeling and so on, like the ones I show you, like the protein structure. So there's a big area by itself already. So it really depends on what you kind of like to do. But I say for beginners, Python, NCBI, BLAST, some familiarity, those kind of things is probably how you started. And then depending on what you want to go further, and then machine learning, for example.
Yeah, I think basically it's just set your destiny, like where you want to go in the end, and that journey, go to the destiny, learn all the skill that you think is necessary.
Just start from basics. Start with some basics Python, and then see how it goes, and what kind of career that you like to do. It's a bit hard because the field is evolving as well every day, so it's hard to see what you know now.
True.
For the future.
Another question, for people who are keen to advance their research career abroad, would you recommend doing a master in Malaysia or abroad to be more competitive in securing a scholarship for PhD, or securing postdoc positions abroad upon completion of a Malaysia PhD? Basically asking, would you think like, is it necessary to have a master or like PhD titles if they want to secure a career or overboard?
I think it really, there's no easy answer as well. It really depends. Like, so I know at least from the program that we had in the PhD program. So I think they usually, it's a very competitive program. They take only 40 students, I think, for each intake from around the world. So I think the main thing is they probably expect a master's at that time. So I don't think they care too much about a master's. But I think the PI, usually they will like to see what kind of work that you've done before. At least you play a role in the paper, or what kind of research you do for your bachelor's or your master's. I think that's probably important. If you can apply for master's overseas, I think sure, why not? You can just go and try for it. But at the end of the day, it's not so important where you do your master's, but what did you do for your master's? I think that's probably more important. Like what kind of research that you do. I think, at least in my case, I think I did phylogenetics today, then I did some time in Barcelona and in Aberdeen as well, a bit more phylogenetics there. So I think that's where I guess my PI got quite interested in what I do, in terms of phylogenetics and evolution. I think that's where I got interested in. So what you do for a master's, I think it's very important. But yeah, if you want to tie to a career advancement again, if you think you want to work I guess maybe it's easier if you want to work in the UK, then you do a master's in the UK. I think that's fine as well if you want to do that. But again, I think master's is probably fine. But if you want to do a PhD again, I wouldn't recommend it if you want just for career advancement. I think it's just easier. Yeah, master's, at least in the UK, it's just a 2 years master's, 1 year master's. The master's is a bit different, I guess, or just like a short master's. So I think if you can get a chance to do a master's in the UK, then why not? If you can go for it. But if you want to do a PhD, I think it's less important where, but what you do for your master's.
Yeah. So basically, the achievement that you did, the work that you did in your studies, that is more attractive to people, to employers to look. Yeah, I think we are almost at end of the times. We are ending in 10:30s. Just looking, let me just check whether there's more questions coming in. Yeah, I think, thank you. Yeah, I think that's all. Thank you, Dr. Jun Hoe, for your sharing. I think a lot of people ask a lot of questions about the career. They're quite interested in how you end up in your positions right now. Yeah, thanks a lot. And for the audience, we have a feedback form. Let me share again. Thank you everyone joining in our Research Insiders with Dr. Jun-Hoe Lee. And this is, in the chat box, you can see our feedback forms, and feel free to fill out the forms and we value your feedbacks. Thank you.
All right, thanks a lot as well. Thanks for inviting me as well, and thanks to everyone else for here. And all the best, you guys.
Thank you.
Bye.
Thanks everyone. Bye.